speaker is Gerhard Schwank from the University of Zurich in Switzerland. He studies clinical translation of genome editing tools with the goal of to cure monogenic diseases. Especially, he works on in vivo targeting of genetic variants using CRISPR technologies. And today, the title of his presentation is Treatment of Genetic Liver Diseases by in vivo genome editing in mice and macaques. Thank you, Gerald. The floor is yours.
Thank you for the introduction. Yes, I will present today our work where we focus on the correction of genetic liver diseases by in vivo genome editing. If we think about treating genetic diseases with genome editing, there's two fundamentally different approaches. So the one is the ex vivo approach, where the idea is to take cells of the patient to then correct the disease-causing mutations ex vivo, either in primary stem cell cultures or in iPS cultures, and then to retransplant the corrected cells to the patient. The other approach is the in vivo genome editing approach, and here the idea is to directly deliver the genome editing tool to the organ of interest in the patient. Now, obviously, both approaches have advantages and disadvantages. With the ex vivo approach, it's of course quite a complex procedure to take out cells, culture them ex vivo, but you have the advantage that you, in principle, could select for the cells where the genome editing tool was corrected the pathogenic mutation and where it was precise and didn't induce off-target mutations. The in vivo approach is more straightforward and less complex in a way, but of course it has to be extremely efficient and precise because you cannot select for the correctly edited cells. Now, when I started my lab now already a couple of years ago, in 2015, we came more from actually the ex vivo genome editing approach, since I did my postdoc in an organoid laboratory. And when we kind of investigated whether this is feasible for genetic liver diseases, we actually figured out that there's quite some bottlenecks, technological bottlenecks still here at the moment. So one is that if we isolate cells from the liver and try to culture and expand them, there is a few technologies. So there's the liver organoids, the chemically induced liver progenitor cells, or the iPSC technology. But actually with all these tools you can expand primary cells, but to then really differentiate them into mature hepatocytes is not yet possible. So with all these systems you get hepatocyte-like cells, which unfortunately don't really perform all the functions, the necessary functions of hepatocytes. So transplanting those into mouse models will not give you a healthy mouse. And also, actually there have been already several clinical trials where not whole organ transplantation was used to treat patients with genetic liver diseases, but hepatocytes were isolated from donor livers and only hepatocytes were transplanted or infused into the portal vein of the patients. And unfortunately, none of these clinical trials, there have been really long-term benefits observed. So also the transplantation of hepatocytes, although there is it's a very regenerative organ, is potentially not as easy and straightforward as one would wish. So overall, for us, for our lab, we kind of thought it's a too hard nut to crack for us, and we kind of then more focused on the in vivo genome editing approach. But with the initial CRISPR-Cas system, which has been already nicely explained in the previous talk, where you have an endonuclease that is targeted to the region of interest with a guide RNA, and then generates a DNA double-strand break, you have the issue that what this system really essentially does, it generates a DNA double-strand break. But then to repair this double-strand break, or to precisely repair genetic mutation, you need to actually specifically change the sequence. And the only way you can do that is that you have to rely on the cell to use homologous recombination and the donor DNA that will be integrated, and then a short sequence stretch can be exchanged. Now, unfortunately, in most cell types, DNA double-strand breaks are not repaired by homologous recombination, but rather by non-homologous end joining pathways or procedures, which are relatively imprecise and therefore often generate small insertions or deletions. So essentially, in most cell types, CRISPR-Cas nucleases are very efficient to generate small deletions and therefore gene knockouts, but it's extremely inefficient to introduce precise DNA changes. Even worse, the homologous recombination pathway is only active during the cell cycle in dividing cells. And the liver, of course, is an organ with a very slow cellular turnover. So in the liver, using regular CRISPR-Cas, actually, especially in adult animals, we have over 99% of the edits will be small deletions and not precise edits. In addition, there have been already several studies showing that CRISPR-Cas-induced DNA double-strand breaks not only lead to small indel mutations, but also quite frequently lead to complex DNA damage, chromosomal rearrangements or large deletions, which activate P53. So you kind of select for cells that are already mutant for P53, and they are also potentially oncogenic. So there's also a kind of a safety issue using CRISPR-Cas nucleases. However, at that point, I have to mention that this is it causes it causes fear that something could happen, but so far in large animals or clinical trials, no oncogenic formation during CRISPR therapies have been observed. So it's really currently studied. Now, unfortunately, the CRISPR-Cas field is a very dynamic field, and in the last years, novel genome editors based on CRISPR-Cas have been developed that modify DNA without generating DNA double-strand breaks and without the requirement of homologous recombination. On the one hand, there are base editors where a deaminase, single-stranded DNA deaminase, is fused to nuclease-impaired Cas9, and with this system, either cytidines can be converted into thymidine, so changing CG base pairs to TA base pairs, or with adenine base editors, adenine can be deaminated into guanine, changing AT base pairs to GC base pairs. More recently, also primers have been developed, and here reverse transcriptase is fused to Cas9, and it takes part of the now prime editing guide RNA as a template which can then be integrated into the DNA locus. So here we have a Cas9 nickase, it only generates one DNA break and not a double-strand break. Now, this is an extremely versatile tool, as you can actually exchange or in principle correct any point mutation, but also small insertion or deletion mutations. Now, the first system, so this was 2016, base editors have been developed. So we, when we kind of figured out that it's very hard to precisely correct mutations with classical CRISPR-Cas, we kind of immediately were very excited when we saw these studies coming out, and we immediately wanted to test them if they work efficiently in vivo in animal models. And there we were lucky that we back then already collaborated with the Children's Hospital in Zurich with Johannes Heberle and with Beat Thüni. And they had a mouse model for phenylketonuria. It has a mutation in the phenylalanine hydroxylase, which leads to an excessive amount of phenylalanine in the blood, which causes damages in the central nervous system. Now, fortunately, this mouse model they had was the pathogenic mutation was a T2C mutation, which is a mutation that can be, in principle, converted or corrected with a cytidine base editor. So we've first, so we have here the disease mutation. We first generated now a cytidine base editor that targets that locus and that converts this cytidine back to thymidine, restoring the healthy or wild-type sequence. Now, in the first study we performed already a while back now, we encapsulated this cytidine base editor into an AAV, into a viral vector that transduces liver cells, and indeed we got about 50% correction, which then led to a decrease in the phenylalanine levels back to physiological levels after a few weeks, and also to a correction of all the other phenotypes the mice normally have. So that was very exciting for us, but it was basically a proof of concept study, because the issue with using AAV viral vectors is that you continuously express then the Cas9 base editor, because it's not a transient system, it's a permanent expression. And of course, that's not really desired to continue expressing a genome editor after the correction is actually done, because that could lead to an accumulation of off-target effects. So at that point, then we collaborated with Drew Weissman from the UPenn and with Acuitas Therapeutics, which were already back then heavily investigating lipid nanoparticle-mediated RNA delivery. Back then it was still a smaller field. Now those are more well known, since exactly these LNPs from Acuitas and the mRNA technology from Drew Weissman is also in the BioNTech Pfizer corona vaccinations. But we did not encapsulate the spark protein. What we did is we actually encapsulated the guide RNA for the base editor and the mRNA encoding for this specific cytidine base editor. And then we injected them systemically into the phenylketonuria mouse model. So these lipid nanoparticles enter the cells via the LDL receptor that is expressed in the liver. So if you inject them systemically, they very efficiently are home to the liver. Now what we see here is that indeed with the mRNA technology you get very transient expression. We're here on a logarithmic scale, so basically after 4 days there's no expression of the guide or mRNA left. Nevertheless, this very transient expression was sufficient to lead to about 15% precise correction of the pathogenic mutation, which was again sufficient to reduce the phenylalanine levels below the therapeutic threshold of 360 micromole per liter. Here, as I said, it usually enters cells by the LDL receptor. Therefore, we have actually very specific editing in the liver and not in other tissues. Now, about the time we obtained these results, there were several other publications appearing showing that base editing could be potentially also dangerous due to guide RNA independent off-target deamination. So what we do is we express the base editor, which is a single-stranded DNA deaminase, and basically from these results the fear came up that this could actually lead to a very high random deamination in treated cells. Either on the RNA, on mRNA, but also on DNA. So what we then did first is we actually kind of repeated and looked, well, are these studies actually true? And we overexpressed also our cytidine deaminase. We transfected into HEK cells as done in the other studies. And indeed, when you do that, you see actually tens of thousands of C2T deaminations or C2U deaminations on RNA. However, when we then analyzed our treated animals directly after the lipid nanoparticle needed delivery or a month later, we hardly saw any off-target deamination, which was initially a surprise. Kind of became clear when we looked actually at the expression levels of the base editor. When you do the experiment in HEK cells, where you just transfect a plasmid that expresses the editor, it's 150,000 reads per million reads, so 15% of the whole transcriptome in this experiment was the base editor, and here it's only about 100 reads per million if we do it in vivo. So kind of our conclusion is that with the in vivo approach, we get a much lower or much more physiological expression of the base editor, which is high enough to edit the on-target but low enough to not lead to excessive off-target elimination. So that was on RNA. Of course, we also looked on DNA. There we isolated hepatocytes from treated animals, expand them clonally, and then we analyzed each of these clonally expanded hepatocyte clones that had on-target editing, but actually in those clones, the C to T conversions in the whole genome were not above the controls. And also we did not see any ApoBac signature, which ApoBac is the cytidine deaminase used in our base editor. So from these results, we kind of concluded that we do not induce a massive amount, at least not a massive amount of off-target terminations. The detection limit was about 20 to 50 mutations per cell. So we could still might induce a few mutations, but not too many. And at that point, it's always important to mention that the mutation rate in humans is quite high. So in an adult, the point mutations in hepatocytes are already about 1,000 per hepatocyte, and we have billions of hepatocytes. So we kind of believe that we are here with genome editing in a off-target editing rate that we think should be tolerated also in humans. Now that was all for cytidine base editing and cytidine base editors. We of course also wanted to test whether adenine base editing works with a similar efficiency, also because the majority of pathogenic mutations are actually mutations that are in principle correctable with adenine base editors. Then we turned to targeting PCSK9, which is a negative regulator of the LDL receptor. And it was for us interesting because on the one hand, editing PCSK9 can be easily detected in the serum, either by looking at serum levels of PCSK9 or at blood cholesterol. But it has also therapeutic potential because inactivating PCSK9 leads via regulating the LDL receptor to a lowering of blood cholesterol. So in principle, it could be also a potential therapy for hypercholesterolemia. What we did is we designed base editors that introduced splice mutations in PCSK9, inactivating the expression of PCSK9. Again, we had a transient approach, so we produced the base editor and the guide RNAs as RNA and encapsulated them into lipid nanoparticles, which leads to a very transient expression of the base editor. And then injected into mice, we got about 60% editing in the liver. Here it's also important to mention this is whole liver. If we isolate hepatocytes and only analyze hepatocytes, we are at a range of above 90% of correction or of editing. Now, as expected, this really led to a significant decrease of plasma PCSK9 and also of plasma LDL levels. Next, of course, we again checked for off-target elimination, either on RNA, and there again, if you do that in vitro in HEK cells, you have tens of thousands of A to T conversions, A to G conversions on mRNA, but in vivo, either 48 hours or 17 days after LNP injection, we only saw a very limited amount of off-target elimination on mRNA. And likewise, also when we again analyzed hepatocytes isolated from treated animals, we did not see an increase in A to G mutations in the genome of these hepatocytes. And we also did not see a signature, mutational signature for TED-A, which is the deaminase used for adenine base editing. Now, the nice thing about targeting PCSK9 was also that it's a wild-type gene. We targeted wild-type gene, so one does not need to generate transgenic animal models to test that system. And that actually allowed us, together with Aquitas Therapeutics, to test that approach into the system in non-human primates. So in our study design, again, we encapsulated a base editor now targeting the primate PCSK9, again introducing a splice mutation in exon 1 into lipid nanoparticles, which were delivered via systemic injection into non-human primates, and then we analyzed them a month after the treatment. So this led to editing rates, depending on the dose, of up to about 30%. Actually, these editing rates were very consistent to our mouse experiments, because in mice, when we used a dose of 1.5 mg per kg, we also got about 30% editing. This again led to a significant reduction in serum PCSK9 and also in serum LDL levels, showing that in principle the LNP-mediated mRNA delivery of base editors allows to edit the genomes also in primates. Now, I'm aware that the reduction of serum LDL levels is not too impressive here, but actually in independent studies performed per Verve Therapeutics, which now I think also entered the clinical trials, they now even optimize the approach and get higher editing, and as I said, are now entering the clinics with the idea of targeting PCSK9 to treat familial hypercholesterolemia. So of course we also looked at editing at other tissues. Interestingly, we found some editing in the spleen, potentially due to the uptake of the LNPs also of dendritic cells. And what is also not a surprise, we actually do see the initiation of an adaptive immune response to the base editors, which are bacterial proteins, about a month after the treatment. Now, this must not be necessarily a problem, because on the one hand, you probably can do a single-shot therapy, and even after redosing, you inject the RNA and LNP, so neutralizing antibodies against Cas9 or TED against the base editors shouldn't be an issue. But of course, the cells that are then transduced and express the base editors might get attacked by an adaptive immune response during redosing. But this is, several groups are working on that, and I think it looks quite promising that this is not really happening. Now, of course, when we do base editing, there's a limited targeting scope, because we cannot correct or introduce any DNA change. So it's basically transition point mutations which we can convert. Now, if you look at in the CleanVar data set, about 60% are single point mutations of pathogenic mutations, and about, again, 60% of the single point mutations are transition point mutations that are in principle correctable with base editing. So overall, we are at about 35–40% of pathogenic mutations are theoretically targetable. Now, what about the other 60%? And here, other technologies are needed. And as I said already before, more recently in 2019, a new technology, prime editing, has been developed also in the LIU lab at the MIT, where a reverse transcriptase reads a designed RNA sequence attached to the guide RNA. And with this, one can in principle correct any small genetic change. So transition, transversion mutations, but also small deletions and insertions. Now, this has obviously a very high potential, and when this system was published, of course, we and others had interest to assess whether this system also works in vivo in the liver. Now, when we looked into that, the first issue we had is the large size of prime editors, because on the one hand, Cas9 is large already, but also the reverse transcriptase fused to Cas9 is a large protein. So we first generated deletion mutations and screened whether we can actually remove domains of the reverse transcriptase. And indeed, in one variant where we removed the RNase H domain, we got a very similar editing on two different reporters, or identical editing, and also on different loci, endogenous loci we tested. So with this, we could already at least reduce it to about 10% in size, the prime editor, which is still large, so it's still about a 6 kb the reading frame. And so when we now wanted to go in vivo, we first tested, of course, again, the viral approach, similar to base editing. And our first idea was also to use AAV vectors again, so adeno-associated viral vectors, since these are the least immunogenic viral vectors for delivery. Now, these have the only big restriction that the packaging capacity is very limited. So we kind of had to first split this prime editor in 2 parts using a transplicing system. It's an INTIN transplicing system, where basically we then expressed the N-terminal part from one AAV and the C-terminal part from another AAV. So with this system, it just fitted on 2 AAVs. And what we then first did is we introduced a silent G to C mutation in the DNMT1 locus. Again in the liver. So we encapsulated this prime editor into an AAV vector, and we injected them either into newborn animals systemically or into adult animals. And what we observed here is that in particular newborn animals, the editing rates were actually quite decent, about 15%. In principle, that would be enough for a large number of genetic or metabolic liver diseases. In the other adult animals, however, the editing was quite a lot lower. So we thought it could be either because cells need to proliferate for efficient prime editing, similar than with normal Cas9 nucleases, or because of the higher relative dose, because we injected basically always the maximum volume, which was led to a higher relative dose in newborns compared to adults. So to test the proliferation, we then again injected adult animals and performed partial hepatectomy to induce hepatocyte proliferation, but that did not increase editing rates. And then what we did next was that we reduced the dose in newborns to get to the same relative dose than in adults, and then indeed we got the same editing rates in newborns and in adults when we injected the same number of viral copies per kilogram body weight. So that also led us to speculate that we could probably boost the editing rates if we further boost the expression of the prime editor. And since these INTIN split systems are not so efficient, we've then turned into an adenoviral vector, which has the advantage that it has a larger packaging capacity, so we could package the full-length prime editor, but it has the disadvantage that it's more immunogenic, so the clinical translatability is limited. Nevertheless, we saw that with this system, we really then got 60% editing in adults and 40% in newborns, so that was actually very promising. And last but not least, I told you that it's a very versatile genome editing tool. So of course, in principle, you can also correct the pathogenic mutation in the PKU mouse model, and that we then also did. And with actually the most efficient prime editing guide RNA and prime editing combination, we got in newborn animals editing of about 10 to 12%, which again led to a reduction of blood phenylalanine levels to physiological levels. And what I still want to mention finally is that we think one big advantage of prime editing is that it's really highly precise. The reason for that, and this kind of data from our study, but also a lot of other people already studied that, and the reason is that on the one hand, in prime editing, Cas9 needs to bind to DNA to nick the DNA, but then you also have this template for the reverse transcriptase, and if it just binds an off-target site, usually this template for the reverse transcriptase does not fit to the locus. So that's why actually we and others have so far not really detected any off-target editing with prime editing. And because you can really design the template, you also don't have bystander mutations, meaning mutations just next to your mutation you want to correct, which is different with base editing, where often when you have a cytidine you want to correct, there's another cytidine next to it which you do not want to edit. And that's, for example, also what happened in our mouse model, that with base editing we always have quite some loci, then we introduce another mutation just next to the corrected mutation. And this is really also not an issue we have with prime editing. So in conclusion, we think it has a lot of potential, prime editing, but so far we still rely on viral vector delivery to get efficient editing in animal models. And with that, I'd like to thank you for your attention, and I'm happy to take questions.
Thank you. Unfortunately, we only have very little time for questions. Are there any questions from the audience? Otherwise, I would start with some.